All right, welcome. So I imagine a couple more people are going to join us. I'm just going to let them take a second to roll in and then we'll get started. While we're waiting, go ahead and put your info into the chat. All right, well, let's just get started. So typically how these lab meetings go is we pretty much do get started on time, and usually I will open up the lab meeting a few minutes early so that you can get settled. I'm sorry today was a bit disrupted, so we start late today. And hopefully everyone got that memo. All right, we're gonna do a quick AV check. So right now your microphone is probably muted, and that's what the default is set to be. I'm just gonna call the names as I see them pop up in the chat. And when I call your name, go ahead and unmute and just say, hi, my name is, so that I can hear how to pronounce your name. And then remute when we're done. So let's start with Marina.
Hi, I'm Marina.
Hey, Marina, good to see you. And Samba, go ahead and unmute and say your name.
Hi, I'm Samba.
Samba, thank you. All right, it looks like we have another person in, but maybe, I'm not sure if the microphone is working or not. Preet, are you able to unmute and say hello? It's not showing me an option for your microphone, so I kind of wonder if the microphone is not working. Let me just all right. So these meetings will be recorded and posted, but it's a great idea to come to them, especially because there's time to ask questions, so they can be a little more interactive. Okay. Check that you have. Okay. All right, so typical lab schedule. The lab prep is due Thursday, and what that means is that there's a background and introduction pages that you want to read in order to complete the pre-lab, which is due on Thursdays. Then you'll also post in the Lab 1 discussion by Thursday to tell your group which part of the collaborative part of lab you'll do. And then Sunday is when the labs are due typically. So there's going to be data or tables or something of that nature. And if some of these deliverables are ones that you've worked on together, it's fine for them to be the same. If it's officially part of the group work, if it's an independent activity, it's fine for you to ask for help and talk it over with your lab mates, but make sure that your work is originally yours. Also, you're going to do a conclusion for lab. And so make sure to get all of the items into the Dropbox that are asked for. We have a post-lab quiz that's due also on Sundays. All right, so just to check in where you should be at already, I am going to finish making lab groups at the end of today. So today is the last day that you can choose your own lab groups, and by tomorrow morning, I will have assigned everybody who's left over. You need your lab safety quiz done by then. That's mainly helping you to make sure you're safe when handling the home lab kit. And then the preparation for lab 1 is due Thursday. Just because this is the first week and there was a holiday, I will actually keep the pre-lab quiz open until the end of day Friday and accept it late. It's just in case you have scheduling issues, but in the future you want to be done by Thursday. So again, that needs means you read these introduction and background pages, use that to submit the pre-lab quiz, and post in the Lab 1 discussion, just saying, this is the thing I'm going to do. Okay. One other thing I want to mention is that the pre-lab quiz has unlimited attempts until the deadline, so you can retake it. And the post-lab quiz has 1 attempt only. Most of the labs for the first couple of weeks are virtual, and Lab 4 and 5 are also virtual. But there are 2 labs that use your lab kit. That's Lab 3 and Lab 6. And so the virtual labs follow the timeline that we've just talked about. That is to say that in the week they're assigned, the pre-lab is due Thursday, as well as the discussion post where you say what you will do. And by Saturday, you want to come back and say what you have done. And Sunday, you want to deliver all the things. But Lab 3 and 6 have extra time because they require you to grow stuff and it takes time. So you actually get 2 weeks for this one. Now, because we don't use the lab kit for the first 2 weeks, if you had to delay purchasing it for some reason, you can do that, but I advise you to buy it by next Monday so that you are not behind on Lab 3. So this is just a suggested timeline for how to tackle all this stuff. Like early in the week, you want to do the background reading, make sure that you do the pre-lab quiz on time and post in the lab discussion. By now you'd know what you're supposed to post. And then the next couple of days actually do the stuff and then submit on Sunday. Online, you will have seen how to order the lab kit. And so here's the website again. You want to make sure you're looking for the Everett micro kit, not the Cell Bio kit. The Cell Bio kit is cheaper and so it's appealing, but it's the wrong one. So you need to pay about $90 for this kit. When you put in your info, you get to choose if you want it shipped to you, which is an additional cost, or if you want to pick it up, which will require that you can drive to Monroe. If it's shipped, it should ship out in about 2 days if you're local, but it can take longer if you're further away. And we do, as I said, use this kit in week 3. I do want to do a quick check. There's a lot of noise in my area right now. Can you guys hear me above the background noise? Is it super annoying? Just write into the chat. Okay, great. At any time, if something is going on in the Zoom that's making it hard for you to understand, like something in the background is too loud, or you missed something that I said, or sometimes I think that I'm screen sharing and it's not the right thing, please do speak up in the chat and let me know. I'm happy to adjust those things. Zoom all kinds of things can happen. With that said, give me 1 second. I'm just going to close a window here because it is awfully loud. All right, thanks for your patience. Okay. Instead of doing a breakout, normally I would do a breakout, but we have a small group. So we're just going to do a really quick introduction, you're going to basically just unmute when I call on you and just say a couple things about yourself and whatever comes to mind. So think about what you'd like to share and I'll start so that you know kind of what to expect. So I'm Hillary. I'm your instructor. You can call me Dr. Kemp, you can call me Dr. Hill, you can call me Hillary. All of those things are fine. You can call me Professor. And in addition to teaching and doing research, I am a partner dancer. So I teach salsa dancing in downtown Everett. All right, so I'll give you a second to think about what you want to share with a small group of people here. And let's start with Marina. Go ahead and unmute and just say a couple things about yourself.
Okay, hi guys. I'm Marina again, and I enjoy playing volleyball. I actually used to play volleyball at EVCC. Last year, I didn't play this year because of COVID, but yeah.
Awesome, yay. Volleyball is the best. Okay, thank you for sharing that. Samba, would you like to unmute and share something about yourself?
I'm Samba again. I enjoy playing soccer and going for runs. That's about it.
Love soccer. I used to play terribly, by the way, but I absolutely adore that game. Okay. And Manpreet, are you able to unmute and check in? Is your, go ahead.
Hi, my name is Manpreet and my major is nursing. This is my last class that is required and I'm applying for nursing school for winter quarter. And I love hiking, dancing, pretty much that's all.
Excellent. All right. Thanks so much, guys. That's fun. Okay. All right. So let's talk quickly about what you're actually doing in this week's lab. I want to share where stuff is on Canvas and kind of walk through it. So I'll do that in a second. I'm going to sort of walk through this slide first. The pre-lab, again, you want to read both the background and instruction pages. They're separate. You will need to read them both so you can completely answer the questions. If you're having trouble answering these questions, feel free to reach out to me. The lab one discussion is where you're going to post what you will do for group work. Okay. And so basically the collaboration aspect of it is one where by Thursday you'll just say, hey, this is the part I'm doing. And then by Saturday you'll come back and provide the answer. Give me 1 second, guys. I just have to take care of some. All right, sorry about that, guys. This is the danger of working from home. People just assume that you're available. Okay. So the lab one activities are 2 activities. There's a research part where instead of physically growing stuff, which is, of different places in your home, which is not really safe to do at home, you're going to research different species and where they live. And so you as an individual are going to find the name of 5 particular species of microbe and share them with your group. And as a whole, then there's going to be like 20 things that you'll have learned about. You're not going to be tested on these, but this gives you a sense of what's out there. The other part of it is you're going to do some calculations. So this is more descriptive, like who's living out in your world. And this is more quantitative. If we want to assess how much microbial content there is in air and water samples, there's a way to calculate that. So I'm going to walk through that with you today. All right. So first, oh, you can't hear me. Is that still true? Can everyone else hear me? Okay. Alex, try adjusting your. Or before we do this, actually, I'm so sorry. I forgot to go to Canvas. I wanted to share with you where everything is so that you actually are not at all confused. So this is the home page. In this course, I really want you to actually, from your landing pad, I want you to go to modules. And this is where everything is going to be located that you need. So of course, right now you're doing the orientation module that has to be finished by tomorrow. Here's the week 1 content module, and here's lab 1. Okay, so here's the link that I was mentioning where you have to read about this lab, like some background information. There's usually some text to read and some videos to watch. And then here's the actual instructions for what the activities are. So I did want to walk through this with you really quick. Okay. So this first part, which is a research microbial survey, you're going to do with your lab partners. And so there are different environments that you're going to research sample. And so you are going to look up a species that's in a particular location, like something that's growing on a plant or animal, or something that's growing in soil or water, or something that's growing on some kind of surface in your home, or something you'd expect to find on a personal possession. You're just going to choose one of these areas. And then on in the group discussion, you're going to say which of those areas you're going to look at, and other people will look at different places. And then what you're going to do is actually Google around to try to figure out what microbes grow in these areas. And you need to find a species name of one microbe that can be found in this location. And then by Saturday, you'll come back and tell people, this is what I found that grows in soil, or this is what I found grows on giraffes, or whatever. So here's an example of how you would answer this part of the prompt. You would say, I chose to look at what grows on my cell phone, and it turns out that you can find this bacteria, Proteus mirabilis, there. The second category is microbes that live on you, so your normal microbiota. And here are some areas of your body that normal microbes grow: skin, large intestine, mouth, upper airway. So you're going to pick one of these, and you're going to say in the lab discussion by Friday, you're going to say, I'm going to look up what grows on human skin, or I'm going to look up what grows in our mouth. And then you just got to go and research and find a particular species that lives normally in people in this area, and then come back and say, this is what I found. Okay. Now here are a bunch of processes that microbes are used in. And so you're going to pick one of these. Again, you're going to post and say, I pick making yogurt, or I pick biofuel production, or whatever it might be. And then you're going to look up what microbe or microbes are involved. So just need the name of one particular species. Okay. Same with this prompt. So these are all areas in the biosphere that microbes are important. So you're going to pick one of these and say what it is, and then you're going to find one species that's involved. And finally, diseases. Oh my gosh, so many diseases, right? So here are just some ideas, but you can, if there's your favorite disease is not here and it is microbial in origin, you can do something else. But you're going to post and say, this is what I'm focusing on, and then find that one species and share it with your group by Saturday. And then that is what it's going to allow you to fill out a table that looks like this. You can reformat this table if you want, but all the information that's here needs to be here. And this is just a sample, so you know kind of what it would look like. You're going to have 5 entries, 1, 2, 3, 4, 5, and you're going to say these are mine, and other people are going to have 5 entries in different spots. This annotation part has to be there. Before I move on to the CFUs, does anyone have any questions about how to do this part of the lab? If you do have questions, because we're such a small group, you could just unmute and ask them. Normally I use the raise your hand option, but we are a tiny group. So if you have a question, go ahead and just unmute and ask it.
Are you always going to give us options to choose from on what we research? Like, for example, I said at the bottom, like I was said phone screen or something like that. Would there always be options that we choose from to pick one from?
It depends. If it's a collaboration, then there's going to be options in terms of which part of the collaboration you do, and then you'll tell the people, this is the part I want to do, and other people will do different parts. For some of the independent activities, there'll be choices too, but not always. So for example, we'll see a really good example of an activity where there's no choice. And that's the one that is the second part of Lab 1. And that's where you're going to do these quantities from pictures that I provide. So you don't get to choose different pictures. That you use the pictures I show, and then you do the math based on those pictures. Does that help or make sense, Samba?
Yeah.
Awesome. Great question. Any other questions, guys, before we talk about the next part? Great. Okay. I'm going to go back to the slides for a second. Second part of lab involves quantification, and I'm going to look again with you on Canvas in a second so you can see the pictures, but I want to talk about them first. So there's microbes all over the place, but we can quantify, we can count the microbial load in air and water samples. It's not a perfect metric, but we can do a crude job of it and can use it to sort of compare between areas. When we are quantifying the microbial load in air, we typically measure how much stuff lands in a particular area in a particular time. And so this is one standard unit. It's very common to count how much stuff lands in a square foot, that's the area, in 1 hour, which is the time. So we call this CFUs per square foot hour. CFU stands for colony forming unit. That means something falls out of the air that is capable of growing on whatever medium you're using, whatever food you're giving. And it grows fast enough that it goes from being invisible, one cell is not visible, to being a mound of clones. It basically just replicates and clones itself until that mound is visible to the eye. and that amount is called a colony. And so we're measuring like colony forming units, meaning the stuff that falls out of the air that can actually grow on this food, because we're going to miss some things. So how we do this sample is we take a Petri dish of whatever size, we open it up to the air, and we just let stuff land on it for a particular amount of time. The simplest way to do this, of course, would be to take a Petri dish that was a square foot in size and open it for an hour. But actually that's expensive and time consuming, and we're lazy and cheap. So we usually use a Petri dish that's smaller, and we usually expose for a shorter period of time, and then we just extrapolate out. So for example, if we were to expose a plate for 15 minutes, what fraction of an hour is that? Put that into the chat. Just write into the chat what fraction of an hour 15 minutes is. And if you're not sure, you're always welcome to use a question mark, but I will wait for everybody to reply. Okay, so we want to know how much stuff would fall in a whole hour, but we're only exposing for a quarter of an hour. So let's suppose we had a plate and it was a square foot in size, and we exposed it for 15 minutes and 7 things landed and grew. How much would have landed in a full hour? And how do we calculate that? We had 7 things land in a quarter of an hour. How much would have landed in a full hour? Yes, Marina is correct. Marina, would you like to unmute and tell us how you got that answer?
Yeah, so you said 7 things landed in 15 minutes or a quarter of an hour. So I multiplied 7 by 4.
Yeah, exactly.
Okay.
Yeah, so we can take the amount and we can multiply by how many sort of units we have to account for. And I'll show you sort of the math. You can do the math 2 ways. There's an intuitive way and a mathematical way. Now, often we don't use a plate that's a whole square foot. We usually use a plate that's smaller. For example, a standard plate is 10 centimeters, and that's a 15th of a square foot. So let's suppose those 7 things landed, and we needed to convert just to account for the area. Just to account for the area. What do you do to that number 7? Because our plate is much smaller than a square foot, but we want to know what would have happened in a whole square foot. If it's a 15th of a square foot, what do we take our 7 and multiply it by? Exactly, Alex, that's right. Multiply it by 15, right? If 7 things fall in a 15th of a square foot, well, you would have had to lay out 15 of those plates to cover a whole square foot. So we take our 7 and we multiply by 15. So if we have a shorter exposure time and a smaller area, we have to take our number, let's say 7 in my example, and we have to multiply by a factor that accounts for both time and area. And so we'll do some of these examples together in a second. Water samples are more straightforward in a way. You don't have to worry about exposure time. You're not worried about area. You're worried about volume. So let's suppose you wanted to know how much stuff was in your drinking water. You could just take a milliliter of drinking water, put it on a plate, spread it out, let it dry, let it grow up and count. And that would give you your CFUs per mil. Okay, so then you would know how much stuff was in a milliliter that could actually grow under these conditions. And so if 7 things grew, and after putting a mil on a plate, then you know it's 7 per mil. However, most sources of water have more microbes than would make that simple. It's not, you're not going to get 7. You might get 7,000 and you're never going to be able to count that. So instead what we do is we usually use a fraction of a mil. Instead of a whole mil, we quite often use 0.1 mils because that amount will dry on a plate in a reasonable amount of time. So if I had 7 on a plate and I only put 0.1 milliliters on it, what would have been in the whole milliliter? And how do I figure that out? If a tenth of a milliliter has 7 things, how much does a whole milliliter have? Any thoughts, guys? And if you're not sure, just put a question mark into the chat so I know that you're still thinking. Can I repeat the question? Yes, Alex, great. Yes. So let's suppose that we want to get this information. How much stuff is there per milliliter of a water sample, like a pond or drinking water? But we don't sample a whole milliliter because that would take too long to dry and there's too much stuff in it. So we only take a tenth of a milliliter and we put it on the plate and we grow it up and we get 7 colonies. So since we got 7 colonies in a tenth of a milliliter, how many would we have gotten in a full milliliter? Close. Yeah, you're really close. So you're on the right track though. So if we take a tenth of a milliliter and we get 7, we just have to multiply it by 10 or to get 70. Does that make sense? Got it. Okay. Now, to make matters worse, yes, Alex, exactly right. Usually when we take a tenth of a milliliter of something, it's still too dense. There's too much stuff in it. Like there might be 5,000 things in there still because the original maybe had 50,000 things. So in that case, we use a dilution. So we'll take the water sample, we'll dilute it with pure water, which has no microbes in it. And so let's, for example, do a 1 in 100 dilution. So that means we take a mil, we put it in 99. So now it's diluted versus the original concentration. It is now diluted 100-fold. Then we take 0.1 mils of that and put it on a plate. So if we do that and we get 7 things, we have to multiply those 7 things by 100 because we diluted our original stock 100-fold, and by 10 because we only took a fraction of a milliliter. All right, now I'm going to walk you through this demo, but first I want to go back to Canvas so you can see where this is. This is lab activity B. You're going to do this independently. And if you have questions as you're doing it, it's fine for you to talk to your lab mates about it, but just make sure that you know how to do this yourself. Now, you're going to be looking at a picture of a plate that was taken by exposing indoor air. And that's all these different things landed. All these little brown things are one bacterial or fungal cell landing on here and then growing and growing until it formed a mound. But this colony is from one original cell. So each of these round circles is one CFU. So you're going to count them. Now, what you might immediately think about when you see this plate is, oh, shoot, well, what is an accurate count going to be? Because is this 1 colony or 2? Is this 1 colony or 2? Did these like really small dots count? Okay, so you don't have to get a very super precise number. As long as you do this correctly, you'll be in the ballpark and that will be good enough. And so you might get a like, you know, let's say you got 27 and someone else got 26. I mean, that's not a big deal. But you're going to count all of these circles and that's going to be your CFUs. You have a Petri dish that's a 15th of a square foot, so you have to multiply by 15, and you have exposed for 30 minutes, so you have to multiply by 2. We're going to do an example of that in a second. So you're going to come up with the CFUs per square foot per hour. Now, this is a dish that was taken by putting 0.1 milliliters of a 1 in 10 dilution of seawater on a Petri dish. So again, you're going to count up all the circles, and then you're going to convert and say, well, if this many is on 0.1 mils, and then you have to multiply by 10. And if it was diluted 1 in 10 on top of that, you have to multiply by 10 again. Okay. Then you have to fill out this table. And so this table requires you to describe the colonies. So here you're going to have your sample and your next sample. So this is not going to be toilet water. It's going to be seawater and indoor air. This is just an example so you know what I'm talking about. Types and total number. Types are going to be, well, let's start with total number. That's the simplest. Total number is just count all the circles, whatever there is. Types are going to be, you're going to look at this now that you've counted everything and go, well, how many different types are there? Let's suppose the answer in terms of number was 27. Okay, but then you might be like, well, look, these guys all kind of look the same. So that's one type. And these guys kind of look different. So that's a second type. And then there's these guys that maybe look different again. And maybe now you decide there's 5 types or something like that. Okay. You don't, again, have to get this perfect. I'm trying to get you familiar with what the diversity of colonies look like. And then you're going to take any 3 types that you see on there and just describe them in words. Okay. So now let's go back and do the math. And I'm not going to do the math on those pictures. I'm going to do the math on different pictures. So here is an air sample example. Okay. And what I would do first is count up all the colonies. So my animation is going to walk us through that. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 2, 3, 4, 5. Okay, so if we decide there's 25 colonies on here, and I tell you that this plate was 1/15 of a square foot, and that you exposed for half of an hour, then what is the colony number, the CFUs per square foot hour? So remember, you have to take this number and multiply to account for the fact that we only sampled a fraction of a square foot and we only sampled a fraction of an hour. See if you can figure this out and put your answer into the chat. And we'll just give you a couple minutes to think about it, and then I'll walk you through the math myself. So any thoughts, guys? Marina, what did you come up with?
Not sure if this is correct, but I got 93.75.
And tell me how you got there.
I multiplied 25 by 15.
Great.
And then I multiplied that answer by a 4th.
Okay, cool. So the only difference that I would do is instead of multiplying by 4, I would have multiplied by 2 because you exposed for half an hour. And I think you're still thinking about that 15-minute example that we did earlier, but that's the right idea. Yep.
Okay.
Okay. So let me just walk you through the math in this slide. So you're going to have these slides, you can refer back to them. So we have our CFUs, right? And this was achieved by exposing a 15th of a square foot. So we know we have to multiply by 15 because we only took a fraction of a square foot. It would take 15 of these plates to get the right number. And the exposure time was 30 minutes, which is half an hour. And so we know we have to multiply our number by 2 as well, because it would take a double the exposure time to get to a full hour. Now there's 2 ways of doing this. If you're mathematically inclined, you might prefer to do this. 25 CFUs divided by the fraction of the area times the fraction of the time, okay? What's gonna happen is 1/15 times 1/2 is gonna be 1/30. And when you take something and divide it by a fraction, the denominator comes up top, and so you end up going 25 by 30, okay? And that gives you your answer. But doing it this way, you know for sure where all the units are because they have to stay on the bottom. If you're more of an intuitive person, then doing it this way is going to be perfectly fine. You're going to say, I have 25 CFUs. I have to multiply it by 15 for each square foot because I only sampled the 15th of a square foot. And I have to multiply it by 2 because I only sampled for half an hour. Either way, you're going to get this answer, okay? And so you were totally on the right track, Marina, except for one little difference with the time and then possibly a decimal point. All right, now we're going to walk through this again using the same number, but just to see if you understand the concept. Okay, so again, let's suppose we got the same answer, but we had done it differently. We used a bigger plate. so that it was actually a fifth of a square foot, not a 15th, and we exposed it for a shorter period of time, what would be the answer be in this case? Take a second to think about it, write your answer into the chat. All right, Alex, what do you think?
I kind of missed the math part, so I'm not sure.
Okay, absolutely fine. Manpreet, do you have any idea how to solve this? Yes. Manpreet, so instead of, good question. Instead of multiplying by 15, we multiply by 5. And Marina has an answer that is her guess, okay? And Marina, would you like to unmute and tell us how you got that answer?
Yeah, so I'm doing the math how it makes sense in my brain. So I did 25 multiplied by 5, and so that's 125, and then I multiplied that by 0.25 to get.
Oh, great. Okay, so you're so close. And that's why your answer isn't numerically accurate, but you're on the right track. So you did the right thing by multiplying by 5, because the area is 1/5. So you want it's a fifth of a square foot. So you want to multiply by 5 to get to a whole square foot. That's exactly the right thing to do.
Okay.
The exposure time is 1/4 of an hour, right? So again, you want to multiply by 4 to get to a full hour. Does that make sense?
Yes.
Yeah. So all you did was you multiplied by the fraction. If you're going to use the fraction, you can. You just have to divide by it instead.
Okay.
And
Thank
that ends up being the same thing. Okay, so let me again walk you guys through the math. So this is what happens when you take a plate that is 1/5 of a square foot. So it's not a full square foot, it's 1/5. And you get 25 things on it. In order to cover a whole square foot, how many of these plates would you have to expose? You'd have to expose 5 of them. So you take your 25 and multiply by 5. We exposed for 15 minutes. We want to know how much stuff would have fallen in a whole hour. 15 minutes is 1/4 of an hour, so we multiply by 4. Okay, so again, the mathematical way to do this, which I just had mentioned to Marina, is to take this number of CFUs and divide by the fraction of area times the fraction of time. And if you do that, you get 1/20, and then the 20 comes up top. However, intuitively, most people prefer to do this. You take your 25 CFUs, you multiply it by 5 because it takes 5 plates to cover a square foot, because each plate is only a fifth of a square foot. And you multiply by 4 because we only exposed for 1/4 of an hour, but we would have needed to have a 4 times longer exposure to get to a full hour. Either way, the answer is the same. Let me just pause here for a second and see if you have any questions or you want me to go through any of this again. And now is a great time to ask your questions or to sort of say, hey, this is where you lost me, because you're going to have to do this for the lab that's due on Sunday. Okay, so let's do this on a scale of 1 to 5, where 1 is I didn't understand that at all, and 5 is I totally understand how to do this. Just put into the chat what number you would say you're at with this. Okay, Marina's a 4, Alex is a 4. Samba, where would you say you're at? Are you able to unmute Samba? You might have stepped away. Okay, thank you, Manpreet. Okay, so which part of this is still feeling a little fuzzy?
I understand it for the most part. I just get confused on like dividing by 0.25 or multiplying by 4 for like 15 minutes. That's the part that mixes me up a little bit.
Yep, yeah. And so depending on the type of person you are, I'm very visual. So this is how I would do it. but you should only do this if this works for you. I actually draw myself a little picture. I'll have a piece of paper and I'll have like a square and I'll say this is a square foot, and I have my little plate and I go, okay, the plate takes up about, a fifth. So how many of those do I have to have? And then I do the same kind of idea with a time as well. The time might be the harder one because it's not as visual. So I will leave these slides with you. They're going to be posted. And then if you have questions as you're doing this and you're just on one of the examples and you're like, gosh, I got confused here, or I know I don't know what I'm doing, or I think I know what I'm doing, but I just want to double check. You should feel free to email me and just kind of check in. Yeah, the exposure time entry, that's where people get hung up. Yeah. So think about it this way. Let me ask you this question. If this plate was exposed for 10 minutes, and this is what we got on it for 10 minutes, we got 25 CFUs in 10 minutes. How many 10 minutes are there in an hour? That's right.
X.
Exactly. Yeah. So if we get 25 after just 10 minutes, and we know that there's 6 10-minute intervals in an hour, we could just multiply this by 6 and then we would find out how much would have fallen in a full hour. Does that help at all? Yes, and Manpreet, so that you know how to get that 1/6, you would do it this way. You would say, okay, 10 minutes divided by 60 minutes is 1/6. Okay, so now I know that I have to multiply by the thing on the bottom. Great. All right, those are the air samples. And again, if you go away, you think you understand it now, or you do understand it now, and then you go away and you're like, oh shoot, it went fuzzy again. You can reach out to me. Let's talk about the water samples. Arguably, they're more straightforward, but again, they do require the dilution factors where people get hung up. Okay, so I pre-counted this plate for you. 17 CFUs are on it. To get these 17 CFUs, we used 0.1 milliliter of the water sample that was previously diluted 1,000 times. So before I walk through the math, I want to see if you can kind of guesstimate your way to figure out what the original water concentration was in terms of microbes. How much microbe content was there in this water per milliliter if we get 17 after using a fraction of a milliliter and diluting it 1,000-fold? What do you think would be the answer? I have to say that all over again because I forgot. I forgot to actually record that. So let me just go back for a second and we get to see it again. Isn't that exciting? So this is the original one, right? Where we got 17 CFUs on this plate when we took 0.1 mils of a 1 in 1,000 dilution. So we have to multiply, or we have to divide by the product of the fractions. So we either divide by the volume times the dilution, or we multiply by 10 because we took 1/10 of a mil, and multiply by 1,000 because our dilution is 1,000-fold. And either way, we're going to get 170,000 CFUs per mil. And then in this new example, where we got 17 CFUs after taking 0.1 mils of a 1 in a million dilution, we can either take our CFUs divided by the volume times the dilution factor, or we can take 17 CFUs, multiply it by 10 because we know we took 1/10 of a mil, and multiply it by 1,000,000 because we know we diluted it 1,000,000 times. And either way, we get this answer. All right, so before we move forward, does anyone have questions? Let's just take a poll again, like how solid does this feel to you? If you had to do this example, like not this specific one, but a new plate, would you be like, I'm good to go, I'm not good to go, I'm sort of good to go? Just put into the chat like good, solid, not capable at all, or kind of like medium. Okay, great. And since you're in lab meeting and other people aren't, who might not be able to watch this even, you may end up wanting to answer questions from your lab mates if they're struggling. And if that seems like, oh gosh, well, you know, I'm super busy and I don't want to take the time to do that, that's totally your call. But I will just tell you this one little life learning hack that maybe you already know. And that is that when you teach other people how to do something, you really learn it. So if you think that you're, you know, doing them a favor, it's really you doing yourself a favor if you explain to other people how to do this who are struggling. All right. So let's just briefly touch base on what your action items are. Officially Friday, but actually, or officially Thursday, but actually Friday. So the deadline on Canvas is Friday, but you haven't, or oh gosh, I need more coffee, guys. I'm so sorry. The official Canvas deadline is Thursday, but you actually have until Friday to do the prep for Lab 1 because it's been a holiday and it's the first week of class. So make sure you get that pre-Lab 1 quiz in and that post in the Lab 1 discussion that says, here are the areas, the 5 areas I'm going to research for my part 1. Saturday, you want to come back and say, hey, these are the things I found, just so everyone has time to assimilate that information to a shared table. And then by Sunday, you want to submit that table for Lab 1A and table for Lab 1B to the lab Dropbox. You also need to write a conclusion section. I'll show you that before we go. And then take the post-lab 1 quiz. And just a reminder that while the pre-lab is unlimited attempts, the post-lab is 1 attempt only. So do not submit until you're sure of your answers. Also, you have to sign up for an unknown ID number. So I do want to show you just those 2 things on Canvas real quick, where the conclusion information is and where the unknown ID sign up is. Let's scooch on back here. Okay, so first of all, how to write a conclusion. It's the same for every lab. Basically, you're going to answer 3 questions about everything you did. So think about part 1A, what did you do? Part 1B, what did you do? You're going to say, what did you learn how to do technically or procedurally? And you'll just have a statement about that, just so I can look at it and see, yes, you registered what you learned about. Prompt 2, I wish I knew more about, or I feel unsure about thing X. If you don't feel unsure about anything and it was all crystal clear, then write about what you're curious about, what you'd like to know more about. And then finally, prompt 3 is what's the big picture. So for every single thing you did, think about why I assigned that. How does it connect to the bigger pictures and the concepts we're learning in class? So please don't have this answer sound a lot like this answer. Okay, this is like, what did you learn how to do? What did you learn how to calculate? What did you learn how to manipulate? What did you learn how to look up? And this is more like, why do that? How is that important? Okay. If you are looking up species, then you're going to learn, you know, what things are living where. If you are doing these calculations, you're going to learn how to compare, like how much stuff is in different places, that kind of stuff. And you can even go bigger picture than that. You can say, you know, this relates to the fact that microbes are everywhere. And so just kind of try to see the difference between like the skills and the concepts. Okay. One thing about the conclusion is that it is going to require you to write more than a single sentence answer, automatically disqualify you for points on the conclusion if your answer is 1 sentence for any of these prompts. Okay, so do write something thoughtful. It doesn't have to be a novel, but reflect on what you've done. All right, last thing I want to show you, and then we'll open up for questions, is in week 1. At the bottom, you'll see this one here. We have an unknown ID project. It's fully virtual digital. Your first step is just to go to this page, unknown ID sample sign up. It's a wiki page. Click edit, add your name to one of these unclaimed and then state which lab group you're in. Okay. All right. So that's our time, and I'm going to stick around. If you have any questions, please do ask. If you want me to review anything we've talked about, or if you have other questions, even just about the class or something that's not lab related, I'm happy to answer that. But thanks so much for coming today. If you do have a question, just unmute and holler it out.
I have a question about the lab.
Yeah.
So we are choosing only 3 colonies, but for the calculating the CFU, do you want us to count all of the like total, right?
Yeah. So for the calculations, you're going to count everything. And you're going to base it on the number of colonies, not the number of types. So sometimes people get this wrong because they say, okay, there's 27 things, but there's only 3 types. 7 of them are pink and 5 of them are blue. So when you do the math, you want to do it on the total number of colonies that you see, regardless of what type they are. And when you're doing the description, the only reason I say describe 3 things is not because there's only 3 things, but because I don't want you to spend your entire life on it. I just want to see that you can describe 3 things. Does that make sense?
Yes.
Yeah, great question. Thank you for asking that. Thanks, Manpreet. You too. All right. Any other questions, guys? Anything that's bothering you or that you feel like needs clarifying?
Hey, Hillary, I might be dumb, but do we have lecture Zooms or is it just lab Zooms?
That's a great question. It's just optional lab Zooms. Now, the lecture is delivered in pre-recorded lectures that you watch on your own time. And then I'm developing a little bit of a follow-up study video. So there's no interactive live stream for the lecture component. Let me just share my screen real quick because you've asked a really good question. When you go to modules and look at the schedule and just follow along in the schedule, week 1, we are learning this topic. And so when you click on the topic page, you'll find that it says what chapter to read in the textbook. And then there's some slides to look at that go along with these video lectures. You are absolutely required to watch these, take notes on them, and use them to fill out the study guide in order to understand the material. Does that answer your question?
Yeah, it does. Thank you.
Awesome. Any other questions, guys? If not, go ahead and log out. I will stick around until everyone's gone in case you have something that you want to ask that's pops up at the last minute.